we have taken a drug
environment and thermoregulation.
Okay, so the hydroxyurea will be delivered very easily. So that's what our idea. Same thing for oxygen responsive lipid conjugate also. Here we have concentrated on the azo bond. So this azo bond, it will be broken at the low hypoxia level. Okay, so the same thing, and very importantly, you have to do the testing 3D steroids assay. Okay, so these 3D steroids, it will be mimicking. Okay, minus more the validation of the 2D assay. So the same thing we did, and we got the concentration dependent cytotoxicity, one of the hydroxyurea. And the third one is multi-omics. So here you can integrate both your in vitro level as well as your in silico studies also. Okay, so where and all you can go on, you can go to genomics, transcriptomics, proteomics, okay, then epigenomics and metabolomics. Okay, so it all leads to your precise medicine. So one such case study is reactivation of mutated P53 protein, okay, through zinc and tin metal coordinates bearing BRCA1 mimetics or breast cancer therapy. Okay, we all know very clear this P53 protein is a tumor suppressor protein. Okay, so majorly 40 to 50% of the breast cancer, this mutations will be there. This P53 mutation. So our idea is to reactivate the P53. That's what our idea. And here we coupled the zinc also, because the zinc ion is very important for the protein folding of the P53. Okay, so our idea is we wanted to reactivate the P53 protein. We wanted to introduce the zinc metal also, as well as here we have used the BRCA1 mimetics. Okay, the BRCA1 gene. Okay, again the breast cancer patients mutated with this BRCA1 gene. Already our lab has developed this BRCA1 mimetics. Some chemical compounds we developed and we got the patent also. So these are the compounds. Okay, so these are the compounds. The 7-hydroxy 4-methane comarate with thiosemicarbazole. And my student Priya, what she did, she has taken those compounds and she prepared the zinc metal coordinates for the reactivation of P53. Okay, so we all know very well, if the P53 is normal, it goes for cancer cell. If it detects, it goes for DNA damage and senescence successfully repaired. But in case of deficient or mutated P53, the normal process won't happen. It goes for the malignant tumor. That's what our idea. Okay, so we did the normal, the regular protocol, all these in silico studies, and we did pharmacophore modeling. So we have done the BRCA1 mimetics, but we wanted to confirm with the silico study. So we did the pharmacophore modeling, and here we got the pharmacophore hypothesis. 2 ring residues should be there and 1 hydrogen bond acceptor. Okay, so luckily it is compliant with our own compound also. So that's what we did the mutation. So my take home message is you can work on any mutated protein. Okay, and very important thing I forgot to tell is we have chosen these 2 mutations. Okay, or 175H and or 270H. It is majorly present in all the P53 mutated patients where arginine is replaced with histidine in 175 position, in another one arginine is replaced with histidine in 273 position. Okay, then we synthesized the compounds, we characterized, okay, then finally we did, okay, until now there is only one drug that is called as Prima. Prima 1, Prima and Prima 1, they are the known P53 activators, and we have compared our own synthesized compounds with that. Okay, and this is
लेवल नहीं है
then resistance and the relapse detected too late. Okay, then prognosis is static instead of dynamic and personalized. Now, how
इधर लेवल
bring the AI tools for resolving these issues?
उसका लेवल
Yeah.
उसका देख के कर दो
So you can integrate the AI in network mapping, heterogeneity mapping, then early detection and AI resistance prediction, AI response prediction, and of course we have so many
इधर तो लेवल दिख रहा है
molecular modeling tools. Okay,
हाँ ये क्या है लेवल दिख रहा है देखो
these
देखो
approaches I have broadly categorized into three aspects. Okay, how the AI
एज ओके है
can be
देखो
used
एज
in
ओके
the precision oncology. Okay, like starting with diagnosis and treatment as well as the prognosis.
करो अपने हिसाब से
Yeah.
इधर से
So
दिख
if
रहा
you take for diagnosis, the medical imaging, okay, and this the deep learning on MG, MRI, and the PET, that is particle emission tomography, and the whole slide images. Okay, so the AI is exclusively we can use for this diagnosis. So we call it as that is radiomics, pathomics, and genomics, more precise staging and risk prediction. And how this AI can be used in the therapeutics. Okay, so it may be used in the analysis pattern of this TMB, tumor mutational burden, and it always, I am stressing, it accelerates the decision of the oncologist, okay, especially for the critical care support. Okay, it won't replace or it never replace the oncologist. Okay, it increases the speed and accuracy of treatment planning, and it mainly it contours the organs of risk. Okay, organs of risk, and it always tells the adaptive radiotherapy. Okay, during the treatment, okay, based on the requirement. For example, the mutations, the measurable mutations, okay, the biomarkers, it can be quantified. According to that, the therapy can be modified. This is the adaptive radiotherapy. So the AI adjusts the treatment plan course based on real-time changes in tumor size or patient positioning. So at last, it will be, of course, it will be used in the prognosis also. It predicts the treatment response and survival using models that combine the clinical imaging and whatever I have shown in my previous slides, the multi-omics data. So always the models can be embedded into clinical decision support systems to guide targeted therapy and immunotherapy choices at tumor boards. Yeah. And the next 4-5 slides, I will be just giving the tools where you can use for your drug discovery purpose. Okay. So these are all the tools. Okay. Some of the tools which are freely available. Okay. But all the time and quickly moving on. So alpha 4 tool is a very good tool. You can go for the protein modeling and PPI. So now we are using the cytoscape. Okay, so many plugins we are keeping it. Likewise, these are exclusively the system. It will run through this deep learning and ML model. So this is exclusively for protein-protein interaction and molecular modeling. Yeah, it goes on. The list goes on. These are all the different tools. And this is exclusively for omics and transcriptomics. The people, those who are interested, you can go ahead with these tools. And this is exclusively for imaging, histopathology, and biomarker tools. Okay. So these I can tell, these tools are the computational backbone of the precision oncology. Okay. And yeah, and coming to my summary. So my summary is always it modulates the response. It goes in mind, okay? So always the small molecules modulate the protein or the biomarkers which are involved in the particular disease, and little bit further the microenvironment. You can focus the tumor microenvironment where the different factors like pH, hypoxia, ROS you can target, and finally you can go ahead with the multi-omics. Okay. So, and my take-home messages: decode the genome, understand the tumor, identify the right biomarkers. Then, based on that, go for to predict, personalize, and adapt. A strengthens decisions. Again, I am stressing it, not replaces them. Target the right treatment to the right patient at the right time. So, by integrating biomarker-driven insights, AI-powered analytics, and multi-omics intelligence, oncology is evolving toward truly individualized treatment strategies that improve patient outcomes and reshape global cancer care. So now we are moving to the era. Every patient is unique, and now we have to go for their treatment plan also very, very unique. It should be more individualized, not the average or group of his treatment. So with this, I'm concluding my talk and my acknowledgement. I'm coming from the institute. It is owned by the Swamiji. My sincere pranams to His Holiness Swamiji and the entire leadership of the JSS Academy of Higher Education and Research, and the principal and my vice principal is sitting in front of me, JSS College of Pharmacy Ooty, and the works I presented is supported by ACT, RPS, and of course, without my students, I cannot present my work. All these students' work I have presented, PhD scholars as well as my PG students, and my special thanks to the Scientific Services Committee, the chairman, respected Vandana ma'am, for giving me the opportunity. And I have authored a book which is a textbook of computer-aided drug design. It is exclusively designed for the syllabus of Inform CAD subject. All the basic details will be here. And I am coming from JSS College of Pharmacy Ooty, so where we have 7 to 8 working departments. We do lot of research starting from phyto and chemistry, pharmacology, pharmaceutical analysis. So as Madam told, we are welcome for you for collaborate to all of us. Thank you for your patience hearing. Thank you.
Thank you so much for enlightening us right from the starting of the core biomarkers and its translation and their role in precision medicine targeting the cancer. Right, I think quickly I have just two important questions. First thing is the whole world is changing, and our general usage is one size fits all. That's our all our drugs do, and you are now going into the depth by using AI. Right. My first challenge is AI can definitely help us in giving the right pathways, and we have been seeing, and the whole conference is on that. But how can AI predict the tension in a patient? How can AI predict? And at the end of the day, all the drugs, apart from drugs, we humans require sympathy, which is also equivalent to a medicine. So, I mean, just out of the box, I just want you to just throw a light on this.
Definitely, sir. Yeah, that's what I 2 to 3 times I have stressed the word. Here is to accelerate the help towards the oncologist, or accelerate towards the drug discovery scientist or drug development scientist. Of course, it is not replace as you told. Okay, you can't need the empathy as a human
precision medicine is definitely a costly medicine. However, it has got advantages to treat rare diseases, etc., etc. But being in a middle-aged income country, right? How do you think India can support fully the precision medicine? Right. Thank
That's
you.
what I have given the challenges, sir. The
Yeah,
cost,
the
it is
challenges.
very, what we are rightly you told. So I think long way we have to travel towards that. Yeah, we are, what I can say, we are in like, it is too infancy.
Yeah, we are in the
Yeah.
transition state.
Yes, yes, yes.
Thank you so much, and over to my audience to shoot any questions. Yes? Yep. Below it.
Thank you, Dr. Julie, for that wonderful presentation. It's not a question, it's just an add-on comment to what the chairperson has said regarding the cost involved, because whenever we talk about precision medicine or genetics, it's a notion that India is a poor country, and do we have such power or money power to spend on all those things. In my opinion, because we also work in this area, my opinion is myth. In the sense, we all know what is the cost of an MRI, which the patient is spending. And when we spend money for MRI, it's not for one time, right? Multiple times these MRIs are taken, or even for that matter, you take CT scan. Multiple times it is being taken, and it's affordable. We have seen it's all happening. But when it comes to genetics, it's not that multiple time. We are going to do only one time testing. That's the first point. And second point is, once we started doing this in a large scale, once it becomes a routine of the clinical practice, it is a standard of the clinical practice, then automatically the cost of this genetic testing will come down. So that's my comment always when we speak about genetics. Because yes, I do agree that's the perception that we all have. It's going to be very costly, and it's not affordable. Patients will be suffering, and so on and so forth. But that's just an add-on comment that I wanted to give.
Thank you.
Thank you.
Because I think the world knows that the only country which has got complete genetic session is the England, right? And now slowly all the countries are marching towards it.
Thank you, Dr. Juby, for that wonderful overview, and I personally also got to learn. It's again not a question, but a suggestion which I am seeking for you, for all the students who are here, to get them inducted into use of this AI technology. What tools do you suggest? You've given, of course, AlphaFold and other things which are not, it's not within the reach of everyone. But what are the basic tools you would suggest which one should have in an institute or at a college level, so that, you know, we can initiate, those who have not initiated work can initiate?
Yes, ma'am. Actually, previously we are completely into the CAD, conventional, we can tell, conventional CAD tool editor. I think mostly people are knowing molecular docking, molecular dynamics like that. So now it is, you can integrate with ML tool. The very quickly I will tell. If you have a data, I think you will be having a data for any molecular docking data. You'll be having so many rows and so many columns and
So I think one last word. Yesterday we had a wonderful national elocution competition, right? Although there was a change in the hall because it was not feasible. I think we got goosebumps when students were sharing the role of AI, right? And how it can be a part of. So I think I will be sharing that in WhatsApp or something. I think they have done wonderful research on how the transformation is going to happen, and I think we should also. I have learned many things from that. Just to add on, thank you. Please, thank you so much, ma'am, for your wonderful.
Thank you, ma'am. Now I request our chairperson, Dr. Ramesh Alluri sir, to come forward for the felicitation of this.
Now I request Dr. Vandana Patravade ma'am, Dr. N. Udupa sir, former research director and principal, Manipal College of Pharmaceutical Sciences, to felicitate our chairperson, Dr. Ramesh Alluri sir, and I would like to call Dr. Hindustan Abdul Ahad sir, Dr. Rupa Karki ma'am, and Dr. Anguraj sir to join the felicitation. With this, we shall now proceed to the next session. I kindly request all participants to remain seated and continue to extend your cooperation. I respectfully invite Dr. Muthusamy sir, Associate Professor, College of Pharmacy, Madras Medical College, and Professor Dr. R.T. Lohia, Student Director, RTM Nagpur University, as chairpersons to evaluate oral presentations. I would like to call Dr. Hindustan Abdul Ahad sir to welcome the chairpersons.
Recall the names of the oral presentators. Please confirm your presence. Nitin Verma, Sudarshana Bora, Venkadot Kavita, Venkadot Kavita, Sujit Kumar, Kalirajan Rajgopal, Kalirajan Rajgopal, Praveen Sekhar, Melkia D, Himanshu Katede, Geeta M, Geeta M, Pradeep B. Channakalavar, K.R. Prasanna, K.R. Prasanna, Mutola Sakshi, Mutola Sakshi, Dr. Himeruddin. If any other names are present in here, please check in the other halls.